Today, we're going to continue on with our discussion of carbohydrate structure. We're going to finish that up and then move on to lipids and fatty acids. The one thing to say at the outset, now that we've dealt with the structures of individual sugars, glucose, mannose, and galactose, and we've also talked about their glycosidic linkages, now, if you look at the structure of a carbohydrate, each carbohydrate has one anomeric carbon and then a bunch of other OH groups that you could imagine attaching to the anomeric carbon of another sugar. You've got four options. And so there's actually a bewildering array of possible carbohydrates you could make. And to make things even more complicated, synthesis of carbohydrates in biology is not templated. What I mean by that is protein synthesis you get in a specific sequence because of an mRNA transcript that tells the ribosome what to do, and that mRNA transcript comes from an RNA polymerase that reads the DNA sequence and then makes the right RNA. And so the sequence of the DNA and the RNA and the protein is all templated. There's instructions, and it has to happen in a specific order. Carbohydrate synthesis is not templated. There's not like a message that's read to give you the carbohydrate structure. Instead, carbohydrate structure is determined by the relative expression levels of enzymes that catalyze formation of glycosidic linkages and hydrolysis of glycosidic linkages. So a lot of the answers to the questions that you may have today, why do plants make the sugar that way, whereas people make the sugar this way through this linkage, that answer is sometimes going to be, I don't know, it has to do with the expression levels of enzymes. So if I have to fall back to that a lot, I apologize. Some of these things are known, but it's a little bit more specialized in carbohydrate chemistry and biology. So I want to introduce a few disaccharides to you that you may have encountered already in your life, and then we'll talk about some oligosaccharides. We won't be able to do everything that's important, but at least what we talk about will prepare you for other things that you might encounter in future classes. So if we link two glucose molecules together, in which the anomeric carbon of the glucose on the left is in its alpha configuration, and the oxygen that's attached to that anomeric carbon is itself attached to carbon 4 of another sugar. This is like we made an acetal with carbon 1 of this glucose molecule, only we used an alcohol from another sugar. This type of linkage we will call an alpha 1,4 linkage because it's between anomeric carbon 1 and the alcohol on carbon 4 of the next sugar with the anomeric configuration of alpha. Okay? And then we'll draw in the rest of the OH groups necessary for this to be glucose. And this structure, two glucose molecules linked together in an alpha 1,4 linkage, sometimes people will write out that sequence this way, one glucose and another glucose, alpha 1,4 linked. This is called maltose, and it's a hydrolysis product of starch, and you can find it in germinated grains like barley. And one of the interesting features of this carbohydrate is that one of its anomeric carbons is tied up in an acetal, but the other anomeric carbon is a free hemiacetal, which means that this has, it's in equilibrium with an aldehyde open chain form. So the hemiacetal is an aldehyde equivalent. So they will call this a reducing sugar because of a reaction called the Tollens test, where they take silver oxide and some other reagents, and if the sugar has a hemiacetal end, the sugar will actually get oxidized to the carboxylic acid and reduce the silver oxide salt to silver metal and form sort of a silver mirror. We have a demo that I should have brought today. Maybe I'll do it next time, where you can tell whether a sugar has a free hemiacetal end because it will cause metallic silver to plate out of a solution. In contrast, there are other sugars that are not reducing. So, and you'll hear people talk about reducing versus non-reducing sugars, and all that means, likely the folks who are talking about it may not know about hemiacetals. So all reducing sugar means is there's a free hemiacetal that's an aldehyde equivalent. Now, let me give you an example of a non-reducing sugar. In this case, we're going to link a molecule of glucose with the anomeric carbon in alpha configuration, and we're going to link that with fructose, which I'm going to draw here. Now, fructose is a 6-carbon sugar, and a lot of its stereochemistry is exactly the same as glucose. The difference is in fructose, the carbonyl is on carbon 2. So 1, 2, 3, 4, and 5, and then 6. Okay, so this portion, carbon 5, 4, and 3, with stereochemistry as shown here, is exactly the same as in glucose. The difference is now that the anomeric carbon of glucose is forming an acetal with an oxygen that's on the anomeric carbon of fructose. Do you see how this carbon 2 here is bonded to 2 oxygens, and this carbon here, carbon 1, is bonded to 2 oxygens? Do you see how carbon 1 is in an acetal with 2 OR groups, and carbon 2 is in an acetal with 2 OR groups? Only carbons 1 and 2, their acetals share an oxygen. So this sugar has no free hemiacetal, and so it will be called a non-reducing sugar. And this particular example of glucose attached to fructose with this stereochemistry is called sucrose. And I believe this is an alpha 1,2 linkage, though it's not clear, since both of these are anomeric carbons, whether that's the right way to talk about it, so we won't worry about it. Okay, so sucrose is table sugar, and it tastes sweet, but this gives us a chance to talk about what's the deal with sucrose versus fructose. So first of all, if this were just had an OH group here, that would be fructose. And fructose and glucose are important intermediates in the early stages of carbohydrate metabolism. In fact, one of the first 3 steps of glycolysis is to isomerize glucose into fructose. And that sets up the retroaldol reaction that you saw on the most recent exam. But what's the deal with fructose? You've heard about high fructose corn syrup, and then that's led to all kinds of controversy in the media. What's going on there? Well, it turns out fructose tastes sweeter than sucrose, but is smaller, so you get more bang for your buck mass-wise. And so you can take corn syrup, which would just be a concentrated solution of sucrose, and if you hydrolyze this linkage to release some fructose, it's going to taste sweeter. In terms of why the media several years back was talking about how high fructose corn syrup was bad for you, and there was some ensuing legislation in some places about the size of sodas that could be dispensed, which was lampooned hilariously on Parks and Rec. I don't know if you've seen the episode with Leslie Knope and the child-sized cup of soda. But for only a nickel more you get, or the little swallow. Anyway, so we actually talked about this in the first hour, and there was somebody who was majoring in food science. We got any food scientists here? Yeah? Okay, so can I put you on the spot? Is that okay? What's the deal with fructose? Is it worse for you than glucose? Okay. Okay. So likely, in terms of like behavior, it's possible that the sweeter something is, the more you want to consume it. I don't know. So it's possible that if something's high in fructose, that leads you to want more of it. I don't know. Someone during the last class at 9 a.m. said it's probably mostly marketing because things like high and sugar make American consumers uncomfortable. And so you do have to be careful about that as a consumer of food products. Sometimes food products will say something like all natural when there's absolutely no way it could be anything other than all natural. And sometimes they'll say 0 grams of trans fat. But if you actually look up who are allowed to claim 0 grams trans fat, it's like you have to have less than a certain number of grams. So 0 doesn't mean 0, it just means less than a certain amount. Anyway, you have to be a thoughtful consumer. So that's sucrose, and sucrose is a non-reducing sugar. We've talked about before when we learned about imines, how aldehydes can be damaging in biology. So the problem with having a high level of glucose in your blood and having it be unregulated is that glucose is a reducing sugar. It does have an open chain aldehyde form, and that aldehyde will start forming imines with any amino groups in your tissues, in your vascular system, and that can lead to the various health challenges associated with diabetes. So let's talk about a few other sugars. Another one that, and our theme today is not memorizing structures, but rather how structural differences create real-world consequences. So an example is reducing versus non-reducing sugars. The next sort of story is about a sugar called lactose. Lactose is a disaccharide in which you have a molecule of the sugar galactose from the Greek word for milk, same parent as our word for the galaxy or Milky Way. And then that galactose is linked to another carbohydrate that is glucose. And the linkage is a beta 1,4 linkage with glucose. Okay, so your body has enzymes that can hydrolyze the alpha 1,4 linkage. It's called alpha glucosidase. You make this in your stomach, I think, and in your saliva. Most adults, human adults at least, the earliest humans did not express a lot of the enzyme required to hydrolyze this acetal, the beta 1,4 glycosidic linkage. And the enzyme that does that is called beta galactosidase. Now, as you know, mammals nurse their young, and for humans, in the human infant, this enzyme is expressed in the intestinal epithelia so that human infants can get nutrition from lactose in milk. But in the early days of humanity, especially prior to domestication of cows and goats and things that would produce milk, the human adult diet, there was no need for beta galactosidase because humans were not consuming lactose. And so in the original humans, the expression levels of beta galactosidase dropped as the infant grew up. And if you no longer express beta galactosidase and then consume milk, you have what's called lactose intolerance, which is some unpleasant intestinal discomfort, bloating, gas, diarrhea, fun words. There used to be a commercial for, like, what was it, Pepto-Bismol, heartburn, nausea, indigestion, upset stomach, diarrhea. It's really great things to be talking about, right? Anyway, if you don't have this sugar, if you don't have this enzyme to hydrolyze this linkage, what happens is your gut microbiome does, and the bacteria that can hydrolyze and consume that sugar then contribute to the bloating and the other symptoms. So if you're lactose intolerant, you can take, either in pill form or liquid form or whatever, some beta-galactosidase, and that will metabolize, that will hydrolyze this linkage for you, and then your stomach and your intestines know exactly what to do with galactose and glucose, and no symptoms. It turns out that at some point in human history, probably as humans migrated into Europe, and I'm getting this from a book called A Brief History of Everyone Who's Ever Lived, which is really, really fascinating story about human evolution and genetics. But as humans moved into Europe, which was pretty cold, and as animals started to be domesticated, there became an advantage to being able to consume dairy products into adulthood, because you can store cheese and butter a lot longer than you can store meat. And so being able to digest cheese allowed you to get through other periods where food was scarce, at least that's the hypothesis. And so there was a mutation along the way that enabled beta-galactosidase to be expressed more abundantly. And so if your ancestors are mostly from the European area, at least in the recent past, likely you are not lactose intolerant, because you come from an area of the world where it was an advantage to be able to consume dairy products into adulthood. If you're from anywhere else in the world, likely you're lactose intolerant, and it's only because the original humans had no need of consuming milk into adulthood. So it's an interesting remnant of interesting intersection of carbohydrate chemistry and enzymology and anthropology that's kind of interesting. All right, so that's galactose. Let's see, we've done sucrose. Questions so far? Let's see, let me answer the questions that came up during the first class at this point, which was, do we have to memorize all this? And my answer is no. What are you supposed to get from it? I do want you to know the structures of glucose, mannose, and galactose. I do want you to be able to recognize glycosidic linkages and name them. I have drawn sugars before, even more complicated than this, where I've said, tell me what kind of glycosidic linkage that is, and then you have to name it. So you need to be able to do that. And I want you to be familiar with the idea of structure and function, that small changes in structure can result in large consequences. We've just talked about how stereochemistry at one anomeric carbon played a role in the development of played a role in human history and the development of the dairy industry. So let's talk now about starch. A lot of the carbohydrates that are in food that you consume are starches. You've got a couple of different forms of starch. One, the simplest, is linear, and this is called amylose. And amylose is just a long chain of glucose molecules linked by alpha 1,4 linkages. So it's like we drew maltose before, but it's like you take tons of these and link them together in sort of an infinite chain. And we'll try to represent that. Oops, no. We'll try to represent that with these squiggly lines, and then we go in afterwards and fill in all the other OH groups. If you're struggling to draw this, and if I'm going too quick, don't worry, the notes will be available, and you can honestly just make a note to yourself that amylose is a linear chain of glucose linked together by alpha 1,4 glycosidic linkages, and with that information you should have enough to draw it. But amylose is this linear chain, and that's a simple form of starch. If you take amylose and you introduce a branching point, you have something different. So before I move on, any questions about that structure? Yeah.
So when starches or amylose is packed in with itself, how does that structure function?
What consequences does this linkage have for shape? I think amylose adopts sort of a spiral shape. I don't think none of these bonds are particularly rigid, and all of these oxygens can be interacting with water molecules, so it's likely a very flexible chain. Yeah, that's sort of all I have. The thing that makes it so that you can digest starch is these alpha-1,4 linkages. If you introduce a branch point, you have enzymes that will take the OH group on carbon 6 and link it to another glucose molecule in sort of alpha-1,4 orientation. So I'm turning my iPad to the side because I can't draw chairs sideways. So you put, and even that is a really cruddy axial carbon, isn't it? Fortunately, we'll just slide that over. Okay, that's an alpha-1,6 linkage. Now what would be really hard is for me to come back and then draw these OH groups not sideways. Siri couldn't hear what I said. Okay. Hey Siri, purchase 12 gross of toilet paper. Yeah. You ever thought about going into somebody's house and saying, Alexa, purchase 12 reams of whatever. Confirm purchase. Something useless. Anyway. And this particular carbohydrate is called amylopectin. And the branch point creates, you can imagine a structure that's highly branched, lots of different branching points. And this will create, there will be structural consequences to this in terms of the shape and the volume of the carbohydrate. The point is just that if amylopectin is very similar to the way that humans store sugar in our livers, which is in the form of glycogen. Glycogen's just like amylopectin, only there are more alpha-1,6 branches. So amylopectin is just linear amylose chains linked together with alpha-1,6 linkages. And in glycogen, there's just a lot more branch points. Yeah? Yeah, how do we selectively get the alpha connections there? If we were trying to do this in the lab, we would get a mixture of alpha and beta. The stereochemistry really is controlled by the enzyme that makes the linkage happen. That's hand wavy because it's possible that the enzyme structures are known. I just don't know what it is. But presumably, the enzyme holds the next sugar and the first sugar together in such an orientation that when you make the acetalates with the right stereochemistry. And that actually makes sense because you have enzymes that can only hydrolyze the alpha linkage, and enzymes can catalyze forward and reverse reactions. So if an enzyme is specific for hydrolyzing a certain linkage, then it ought to be specific for making that certain linkage too. And you can hydrolyze all of these linkages, as I think mentioned before, with alpha glucosidase. If we were to make, and I'm not going to draw this, a linear polymer of glucose all linked together with beta-1,4 linkages, then we have cellulose, which is a component of plant cell walls or fiber. So again, the theme stereochemistry makes a big difference in function. Okay, there's maybe one or two more sugars to talk about. One is glucosamine. That's something you've probably heard about before. What have you heard about glucosamine? Got nothing? You can buy it in bottles. What do people take glucosamine for? Any ideas? Supposed to be helpful for joint pain and arthritis. Yeah. Glucosamine is a major component of heparin, which is a part of the extracellular matrix. So it's thought that if you take more, you'll make more of that stuff. I don't know how well verified it is. And you do have to be very careful with the supplement industry that tells you you need to spend a lot of money to consume molecules that most of us get enough of in our diets anyway. There's a great clip of the Big Bang Theory where Sheldon is talking to somebody about vitamins and just saying how taking a lot of vitamins is just making really expensive urine, which I think sounds true. Glucosamine puts an amino group. It's glucose, only the carbon 2 has an amino group instead of an OH group. And nature often puts an acetyl group on that amine, and then you have N-acetylglucosamine, or GLICNAC, as the carbohydrate chemists like to talk about it. But that looks like this: nitrogen involved in an amide bond with an acetyl group, and then you've got the rest of the OH groups here. And that change in glucose structure, keep in mind, I'm going to draw just a subunit of the linear cellulose with the beta-1,4 linkage, but that small change giving you, putting an amino group in place of an OH group and then putting an acetyl group on that amine can turn a relatively flexible plant cell wall into something that's much more inflexible. So if you take glucosamine, N-acetylglucosamine molecules and link them together through beta-1,4 linkages, you have a carbohydrate that is called chitin, and this is a major component of the shells of crabs and lobsters. It's also a major component of insect exoskeletons. It's a fairly rigid and protective substance, and it goes crunch when you step on it. Yuck. So anyway, again, the theme is small structural differences can lead to dramatic different functions. And the problem is that we're not necessarily talking about why. And the why will have to do with the intramolecular forces and how individual chains of carbohydrates pack with each other. And all of that's probably beyond the scope of what we want to talk about. Finally, you will encounter in your future N-glycosides. This is where the anomeric carbon of a sugar is linked, instead of to another OR group, is linked to an amino group. So I'm drawing here for you the 5-carbon sugar ribose, and it's involved in an N-glycoside because the anomeric carbon is bonded to 1 oxygen and 1 nitrogen. And I'm going to attempt to draw the structure of the base, the nucleobase adenine. I probably will do it wrong. We will come back to these nucleobases, but this is an N-glycosidic linkage. And I'll make you the guarantee with the amino acids, the carbohydrates, and the nucleobases. If you want to come to my office and challenge me, and if I can't draw it correctly, then you get the points back on the thing that you missed, okay? So if you get the structure of, say, thymine or adenosine wrong, and then you show up at my office and say, quick, no looking, no review, what's the structure of this? And if I don't do it correctly, you can have your points back, okay? Dang, there's going to be a lot of people in my office, aren't there? Okay, so that's going to finish up our carbohydrates. Any last questions there? Yeah?
What was the name of the, it was similar to amylose except it had branch points?
Yeah, with the branch points, it's amylopectin. And you'll hear people talk about pectin. That's actually a component. I don't know if you've ever made jam. Yeah, you got to buy pectin, which I think is just this carbohydrate. And I think the carbohydrate's heavily hydrated and helps the jam thicken up. Anyway, freezer jam is really, really good. I like that. All right, so now we're going to talk about lipids. And I brought some groceries for that process to occur. There's a lot of different kinds of lipids, but we're going to start today with triglycerides and fatty acids. We will talk about the metabolism of these things later. We've already actually talked about how your body hydrolyzes the ester linkage between fatty acids in triglycerides early on in the semester. And so I'm just going to quickly draw a triglyceride, which is a triester involving this 3-carbon alcohol called glycerol. That's where the triglyceride comes from. That's the glycerol portion. And then you've got the fatty acid portion, always even numbers of carbons in length from 14 to 16 to 18 and sometimes more carbons. So now I have to count 2, 4, 6, 8, 10, 12, 14, we'll go 16 and call that good. So this is a 16 carbon saturated fatty acid. These things do have non-systematic names. The names aren't necessarily important, but you'll hear them from time to time. So I brought a bunch of stuff that has triglycerides in it. Here we have canola oil, which is from the canola seed, which I don't know what that is. But if you squeeze it, the oil comes out, and there you go. Here's olive oil from olives. Let's see, what else have we got? Peanut oil. And then Crisco, pure vegetable oil, which says soybean oil. We have, all of these are liquids. We have another plant-based triglyceride. This is called organic, no, sorry, coconut oil. By the way, about that word, molecules with carbon in them are organic. So technically, any product that has carbon in it could be labeled organic. When they call it organic, that's a marketing / food FDA regulated term, which refers to the process of raising the animals / vegetables. It used to be that scientists thought that molecules derived from living creatures had some property that was different from molecules that were not derived from living creatures, like there was some weird life force. And actually, organic chemistry began with a scientist named Friedrich Wöhler demonstrating that you could make urea from two inorganic starting materials, whereas they thought urea was a biomolecule that you couldn't get from any other way. And he wrote this note, which was really kind of funny, tongue-in-cheek. He says, "I can't hold my water, so to speak," which I think was a pun on urine. I can make urea without the need for dogs or kidneys or anything else. So at that point, it was discovered that there's nothing uniquely special about molecules derived from living things. They just have carbon in them. So the term organic is different than how we're using it in organic chemistry, okay? If you take willow bark and you isolate a molecule and you use it as aspirin, and then you take coal tar and make aspirin from it, it's exactly the same thing and does exactly the same stuff. Anyway, end lecture. Here, not the actual lecture, but then soapbox. So this is butter that's been sitting in my office since last year when I did this, and apparently some of the triglycerides have leached out into the surrounding cardboard, which is eerily translucent. Let's stick that there. That's lipids from milk. And then here we have Skippy, which has hydrogenated vegetable oil, cotton seed and soybean oil. And then this is regular, all-natural peanut butter. And if you look at this, unlike Skippy, this has a liquid layer on the top. I don't know if you can see that. I'm sort of shaking it. You see that sloshing around? Yeah. Typically, peanut butter doesn't slosh around, right? This peanut butter does because the oil in it has not been hydrogenated, and it's still a liquid. And we'll talk about that in a little bit. But here are some of our products that you'll encounter that have these types of triglycerides in them. So one thing to know about why some of these are liquids and some are solids simply has to do with the hydrocarbon chains. So there's hydrocarbon chains that have no double bonds, and those are called saturated fatty acids. And then there are hydrocarbon chains that do have double bonds in them, and those are called unsaturated fatty acids. And you probably have learned something about these in 351, though I'm not sure how much. We'll talk about the relationship between number of carbons and melting point in degrees Celsius. I always struggle to put Celsius degrees in context because we live in a Fahrenheit world here in the United States. But 0 is freezing, 25 is room temperature, 37 is body temperature. Higher than that is pretty hot. If you hear that it's in the 40s somewhere Celsius, that means people are sweating and it's terrible, right? So if you're 12 carbons long and fully saturated, you have to heat it up past 100 degrees Fahrenheit for it to become a liquid. 16, even higher, that's 63. 18 is even higher than that. And then 20, you have to heat up to 77 degrees. So some of these are solids, even almost towards boiling. In contrast, if you have double bonds in the backbone, so we're going to write number of carbons, double bonds, their location and stereochemistry, and then melting point, again in degrees Celsius. Compare the 16 carbon fatty acid with one double bond. It starts at carbon 9 and it's in the Z configuration. That melting point is 0. So a 63 degree loss in melting temperature because of the presence of a double bond. The reason for that, here is a 16 carbon saturated fatty acid. The corresponding 16 carbon unsaturated one with a double bond at carbon 9 will look like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, cis double bond, 10, 11, 12, 13, 14, 15, 16. This will be the 16 carbon Z double bond between 9 and 10. That one happens to be called oleic acid, but you don't need to know that term. You may hear it. If you go to 18 carbons with the same double bond present, the melting temperature is only a little bit higher. This is called linoleic acid. If you introduce 2 double bonds, the melting temperature goes down further. So 9 and 12 with a cis configuration, you get minus 12. This is called linolenic acid. And you can then actually have an 18 carbon one with 3 double bonds. And this one is, oh no, what did I do wrong? I'm sorry, it doesn't matter, but it's all the 18 carbons that are oleic, linoleic, and then linolenic. Really doesn't matter, so don't worry about that. The interesting thing about this one is it is an omega-3 fatty acid. And what we mean by that is that if you draw the hydrocarbon chain, again, it's going to be 2, 4, 6, 8, 9, oops, and 10 with a cis double bond there, 10, 11, 12 with a cis double bond there, 12 and 13, 14, 15 with a cis double bond there, 16, 17, and 18. Do you remember how we used to say, like 2 days ago, carbonyl carbon, alpha carbon, beta carbon, and so on? Eventually, we either run out of Greek letters or we forget which Greek letter comes next. And so we give up and we call the last carbon the omega carbon. So the beginning and the end, alpha and omega. And then we start numbering from the end. This is omega 1, this is omega 2, and this is the omega 3 position. So you hear about omega 3 fatty acids in triglycerides. They are ones that have a double bond starting with the third carbon from the end. What have you heard about health benefits of omega 3s? Anybody? Yeah, go ahead.
Helps with like heart problems.
It's supposed to help with heart problems, and I'm not sure how or what the mechanism is, but I think there's at least epidemiological evidence that that's the case. What else?
It's also commonly found in fish oils.
Commonly found in fish oils, okay. Can you think of a reason why fish might not need to have fatty acids that have really low melting points? Go ahead. Oh, you were. Oh, that was a. Okay, got it. That was a. I'm interested gesture. Okay, good. Yeah.
Right, so omega 3 is in reference to it's like the double bonds starting at the third carbon from omega.
Yeah, omega 1, omega 2, and omega 3. Yep. And then you've got a double bond. That omega 3 is involved in double bond with the next carbon. So fish often experience really cold environments, and so depending on the situation, they may need to not have things that they may need their triglycerides to not be totally rock hard and solid at that particular temperature. So that may be one reason why. Anyway, in terms of health benefits, I'm not sure, but if you see products that claim to be enriched in omega 3 fatty acids, that's what they're claiming, that it has triglycerides that are of this kind. One of the weird ones that I never understood was chicken eggs. Sometimes they sell chicken eggs as saying, hey, buy these eggs, they're rich in omega 3 fatty acids. And I'm like, how did that happen? Because I don't think chickens made omega 3 fatty acids. Go ahead.
Well, I mean, if chickens are fed better, then they produce more and they're healthier, so they can give that to their
Okay.
chickens and put that in it. And they do occasionally have to leave the nest, and if they're not sitting on it and warming it, then the chicken will die. So if this is one that can handle those lower temperatures, that
Okay.
would make sense evolutionarily.
Okay, that might make sense if chicken eggs were able to have a little bit more omega 3s in them. I think another thing that they do is they just feed the chickens feed that's enriched in omega 3 fatty acids, and then they pass that on into the eggs. So just be aware of what's marketing and what's not. Be aware of health terms, but if people are advertising omega 3, that's what it's about. All right, I want to give you a sense for the various compositions of these kinds of oils. We won't do all of them. But just to get an idea of, now that we've talked about melting points, why certain fats are solid whereas oils are liquids. And that's the difference between fats and oils. Oils are solid at room temperature, oils are liquid at room temperature, and fats are solid. So we're going to compare lard with peanut oil and olive oil. I learned the word for lard in Spanish on my mission to McAllen, Texas. That's manteca. And back then, you could go to the grocery store, which was called H-E-B. Vamos a la H-E-B. And in these big yellow 5-gallon buckets, it was H-E-B brand manteca. And you needed, lard is animal fat, and you needed that to make the really good tamales. So in case you've never had good tamales made with lard, you can tell because as you set the tamale on a plate or a napkin, this delicious, transparent, reddish grease will sort of filter out of the tamale and saturate the plate that you're on, and that's the lard. And if you wait, it'll get solid, and you can scrape it off and eat it. I don't know. So we're going to write percent saturated fatty acids, and then percent unsaturated fatty acids, and then we're going to do number of carbons, and so you'll see the composition of each one. All right, so for lard, we're going to talk about the stuff that's 14 carbons, 16 carbons, and 18 carbons. And then for unsaturated fatty acids, we'll talk about 18 carbon oleic with 1 double bond and 18 carbon linoleic with 2 double bonds. For lard is about 1% 14 carbon, about 25% 16 carbon saturated, and about 15% 18 carbon saturated, with 50% oleic and 6% linoleic. So still majority unsaturated, but a substantial amount of fully saturated. For olive oil, it's a little bit less on the saturated side, and 80% of the oleic 1 double bond, and then 7% of the 2 double bond variety. Peanut oil is also a liquid at room temperature, though its composition is a little bit different. You've got 18 carbon oleic and 20 carbon linoleic. By the way, these two are the difference between mono unsaturated and polyunsaturated fatty acids. Okay, so you'll see on the label, for example, it says 16 grams of fat, 3 grams of it here in Skippy are saturated, and 0 grams trans fat. Though actually 0 grams trans fat doesn't mean 0 grams, it just means less than the allowed threshold. So I forget what it is, but it's something that's higher, infinitely higher than 0, as anything would be. But so sometimes you'll see the fats in various products separated out by mono and polyunsaturated. All right, so briefly, let's talk about the difference between Skippy, which is sort of solid, and this Adams peanut butter, which has the gross layer of liquid. Anybody want to come up and just take a sip? So the difference is that the peanut oil in the Adams is mostly unsaturated, whereas the peanut oil in Skippy, at least some of it, has been hydrogenated. By that we mean you've taken hydrogen gas over a metal catalyst, and you reduce the double bond to the alkane. And in so doing, you now convert a saturated fatty acid in an unsaturated fatty acid into a saturated one. Now, why on earth would you want to do that, especially given evidence that saturated fats are worse for you than unsaturated ones? And the reason for this is that unsaturated fatty acids, in the presence of oxygen, can undergo a radical allylic oxidation reaction that installs a peroxide at the allylic carbon. And when you do that, the reason that can happen is because of the double bond, because allyl radicals are stabilized by resonance. When you do that, you change the taste and the aroma of oil. And so products with unsaturated fatty acids have shorter shelf lives than those with saturated, because these ones can get oxidized and go rancid. So next time I will tell you the story of the rancid tostada shells of 2016. These are not them. These were purchased more recently than that, but I'll tell you a story about that. And also explain why you should replace your consecrated oil regularly.